Our understanding of cancer has evolved dramatically, moving beyond simple genetic mutations to embrace a more intricate biological landscape. At the forefront of this new frontier is the field of epigenetics. It offers profound insights into how diseases like cancer develop and progress. Epigenetics is the study of heritable changes in gene expression that occur without alterations to the underlying DNA sequence itself. These modifications act like switches, turning genes on or off, or adjusting their volume, without changing the genetic code. They represent an additional layer of information controlling our genome. In the context of cancer, epigenetics has emerged as a critical player, influencing nearly every aspect of tumor development and progression. It helps explain why cells with identical DNA can behave so differently. This dynamic regulation profoundly impacts cellular identity and function. It is crucial to distinguish epigenetic modifications from genetic mutations. Genetic mutations involve permanent changes to the DNA code itself, like a typo in a book. These alterations are fixed and passed down through cell divisions. In contrast, epigenetic modifications are dynamic and reversible. They are more like annotations or bookmarks in the genetic text, guiding how the DNA is read. This inherent reversibility offers a unique opportunity for therapeutic intervention. The dynamic and reversible nature of epigenetic changes makes them attractive targets for novel cancer therapies. By understanding and manipulating these mechanisms, we can potentially reprogram cancer cells. This opens new avenues for treatment where traditional genetic approaches might fall short. Three primary processes underpin the vast majority of epigenetic regulation. These are DNA methylation, histone modifications, and the actions of non-coding RNAs. Each mechanism plays a distinct yet interconnected role in controlling gene expression. DNA methylation is a fundamental epigenetic process involving the addition of a methyl group to a cytosine base in DNA. This typically occurs at CpG sites, which are regions where a cytosine nucleotide is followed by a guanine nucleotide. This chemical tag acts as a signal. The primary effect of DNA methylation is to typically silence gene expression. When promoter regions of genes are heavily methylated, it often prevents transcription factors from binding. This effectively turns the gene off, preventing the production of its corresponding protein. In cancer, aberrant patterns of DNA methylation are remarkably common. Tumor cells often exhibit global hypomethylation, meaning a general decrease in methylation across the genome. This can lead to genomic instability and the activation of undesirable genes. Conversely, specific regions, particularly the promoter regions of tumor suppressor genes, often become hypermethylated. This silencing of tumor suppressor genes removes crucial brakes on cell growth. It allows uncontrolled proliferation, a hallmark of cancer. Histone modifications represent another critical epigenetic mechanism. Histones are proteins around which DNA is wrapped to form chromatin. Modifications to these histone proteins, such as acetylation and methylation, directly influence chromatin structure. Histone acetylation, for instance, typically loosens chromatin structure, making DNA more accessible for transcription. This generally promotes gene expression. Conversely, deacetylation often compacts chromatin, repressing gene activity. Histone methylation can have varied effects depending on the specific amino acid residue and the number of methyl groups added. It can either promote or repress gene expression, influencing whether genes are turned on or off. These modifications are complex and context-dependent. In cancer, dysregulation of histone modifications is a frequent occurrence. Imbalances in enzymes that add or remove these marks contribute to uncontrolled cell growth and division. This disruption alters the normal gene expression patterns essential for healthy cell function. Non-coding RNAs, or ncRNAs, constitute a diverse class of RNA molecules that do not code for proteins. Among these, microRNAs, or miRNAs, are particularly well studied for their epigenetic roles. They are small, single-stranded RNA molecules. MicroRNAs regulate gene expression by binding to messenger RNA, or mRNA, molecules. This binding can lead to the degradation of the mRNA, preventing protein synthesis. Alternatively, it can simply prevent the mRNA from being translated into a protein. This post-transcriptional regulation allows miRNAs to fine-tune the expression of numerous genes simultaneously. They act as master regulators, influencing complex cellular pathways. Their precise control is vital for maintaining cellular homeostasis. In cancer, altered miRNA expression is frequently observed. Some miRNAs act as tumor suppressors, while others function as oncogenes. Their dysregulation contributes to the disruption of cellular pathways critical for cell proliferation, differentiation, and apoptosis. The epigenetic landscape in cancer is incredibly complex, characterized by a dynamic interplay of these various mechanisms. These epigenetic alterations do not act in isolation. Instead, they form an intricate network that collectively drives disease. Crucially, epigenetic changes can either promote or suppress cancer development, depending on the specific genes affected and the cellular context. This duality highlights the delicate balance within the cell. Disruption of this balance can have profound consequences. One major way epigenetics promotes cancer is through the silencing of tumor suppressor genes. This can occur via hypermethylation of their promoter regions or through repressive histone modifications. When these genes are silenced, the natural brakes on cell growth are removed. This removal of growth inhibition leads to uncontrolled proliferation, a hallmark of cancer. Cells divide without proper regulation, accumulating further damage. The loss of these protective mechanisms accelerates tumor formation and progression. Conversely, epigenetic changes can also lead to the activation of oncogenes. These are genes that, when active, promote cell growth and division. For example, hypomethylation in certain regions can inappropriately activate oncogenes. This aberrant activation drives cancer progression by providing continuous growth signals. It pushes cells towards a cancerous phenotype. The combined effect of silenced tumor suppressors and activated oncogenes creates a powerful pro-cancer environment. The rationale for developing epigenetic therapies is rooted in the inherent reversibility of epigenetic modifications. Unlike genetic mutations, which are difficult to correct, epigenetic marks can potentially be erased or rewritten. This offers a unique therapeutic window. The primary goal of these therapies is to reprogram cancer cells back towards a more normal state. By restoring proper gene expression patterns, these treatments aim to halt or reverse cancerous growth. They seek to re-establish cellular control. These therapies specifically target the enzymes responsible for adding or removing epigenetic modifications. By inhibiting or modulating these enzymes, scientists can influence the epigenetic landscape of cancer cells. This precision targeting is key to their mechanism. One class of specific therapeutic approaches involves DNA methyltransferase inhibitors, or DNMT. These drugs, such as azacitidine and decitabine, are used to reverse aberrant DNA methylation patterns. They are incorporated into DNA during replication. By inhibiting DNA methyltransferases, DNMT drugs prevent the addition of methyl groups to DNA. This leads to the reactivation of silenced tumor suppressor genes. Restoring these genes can re-establish control over cell proliferation and differentiation. Another important class of drugs is histone deacetylase inhibitors, or HDACI. These compounds, like varinostat and romidepsin, are used to alter histone modifications. They prevent the removal of acetyl groups from histones. By inhibiting histone deacetylases, HDACI drugs lead to increased histone acetylation. This generally opens up chromatin structure, making genes more accessible for transcription. The effect is to restore normal gene expression patterns, including those of tumor suppressor genes. Despite the promise, the development and application of epigenetic therapies face several current challenges. The complexity of the epigenetic landscape is immense, with countless modifications interacting in intricate ways. This makes precise targeting difficult. Another significant challenge is the potential for off-target effects. Epigenetic enzymes are involved in many normal cellular processes. Inhibiting them broadly can lead to unwanted side effects in healthy cells, limiting therapeutic windows. Moving forward, personalized treatment approaches will be crucial for maximizing the efficacy of epigenetic therapies. Understanding the specific epigenetic alterations in individual patients is essential. This requires advanced diagnostic tools and biomarkers. Tailoring effective treatments based on a patient's unique epigenetic profile will allow for more precise and less toxic interventions. This precision medicine approach promises to optimize outcomes. It moves us closer to truly individualized cancer care. In conclusion, epigenetics provides a crucial and expanding lens for understanding the intricate mechanisms of cancer development and progression. It highlights that cancer is not solely a disease of genetic mutations but also of gene regulation. This understanding is paving the way for innovative cancer therapies that target the reversible nature of epigenetic modifications. These novel treatments offer hope for patients who may not respond to conventional therapies. The potential to reprogram cancer cells back towards a healthy state represents a paradigm shift in oncology. By restoring normal gene function, epigenetic therapies aim to improve patient outcomes and offer new hope in the fight against cancer.